couple of people back. We probably have a bigger virtual audience than usual. A couple of people still listening in their pajamas. So, Diana Chang here from Genentech. Before as we were walking in together, I asked her where she trained or where she was from. She told me she's from Brooklyn, so she's used to snow. And then I said to her, you're probably young enough that you grew up in Brooklyn while it was already cool. When I grew up, Brooklyn was not cool. Brooklyn was where you wanted to leave and get to Manhattan. But now the prices of real estate in Brooklyn are through the roof. In fact, I'm going back to New York in March, and I want to look at a neighborhood in Brooklyn, just rambling here, which you're used to from me already. All right, anyway, this looks like a fascinating presentation, translating asthma genetics to clinical practice. Thanks for coming and braving the snow.
Thanks for the introduction, and thanks for letting me come speak to you today about how we're using asthma genetics and human genetics in general at Genentech. And I'm currently a scientist in the human genetics department in the research division of Genentech. So there's going to be more basic science here, but kind of giving you a hint at kind of what's coming down the pipeline. Disclosures is I'm a current employee of Genentech.
It's not a conflict of interest, is it?
So just to give you an idea of briefly what we're going to speak about today, I'd like to introduce you to genome-wide association studies, or GWAS. Tell you a little bit about how we're using disease risk genetics in target discovery, and then lead into how GWAS have evolved, and now we're starting to use these cohorts that have electronic medical records and sort of the ins and outs of that and what we have to be careful of and what we're looking at as well. So just taking a step back and thinking about susceptibility to disease, if you have a population of individuals, what determines who becomes a patient and who remains healthy? So there's the influence of environment and the influence of genetics. And depending on the disease, the fraction that it is influenced by environment versus genetics can vary. So what we determine as the phenotypic variance, so the variability in who becomes a patient or not, that we can attribute to genetics, we call that heritability. And heritability can be estimated from twin studies, and that's how it was classically done. And then in more recent times, as these GWAS or genome-wide association studies came out, we can actually estimate heritability from those studies as well. And as an example, in 2013, a group at Harvard actually estimated for a number of common diseases the heritability or the fraction of phenotypic variance that we can attribute to genetics from these GWAS. So what I'm showing you here is the fraction of heritability, the fraction of phenotypic variance due to genetics, so h squared on the y-axis. And on the x-axis is a number of different diseases. So we have bipolar disease, cardiovascular, Crohn's disease, hypertension, rheumatoid arthritis, type 1, type 2 diabetes, multiple sclerosis, and ulcerative colitis. And if you just focus on the light blue bar, the rest of the bars are different models that, for how they're estimating heritability. You can see that for all of these diseases, there's a non-zero heritability component, meaning that even if it's quite small, genetics does influence disease risk. And if we look at the estimates for asthma and allergic diseases, it's about at 30%. So we have a non-genetic contribution to risk of asthma and allergic diseases as well. So genetics has evolved quite a bit over recent years. So, we all started with Mendelian genetics, looking at how alleles combine to determine phenotype. So, for example, in this example, the two little B alleles are combining to produce a different color petal. So if we go back in time to 2001, the cost of getting just one genome was huge. It was the Genome Project, and it was $100 million to get just one genome. And due to technological advances, this has drastically reduced the cost of getting genetics on an individual. And so now in 2019, it's only about $1,000 to sequence a whole genome, which is crazy. So what we often do is compare it to Moore's law, the idea that you can double computation or processing power every year. It's beat that law quite a bit. And so this reduction in cost has not only allowed us to do a lot more research, develop the field of GWAS, but I don't know if you've seen ads, but like a lot of these direct-to-consumer kits are now becoming available. And I thought an interesting tidbit that I read about was that there's now over 26 million individuals captured by these direct-to-consumer kits. Yeah.
Sort of a funny play on words because all these criminals are getting picked up by these kits. You say 26 million are captured.
Oh.
Seems like a lot of crooks.
Yeah, it's actually really interesting if you read about like the ethical implications of these kits and like the I guess the forensics of it all. So I think there's interesting like legality or in the legal world like what we can do about all this as well. But we're not going to get into that. We're going to focus on the science.
Well, if it's still $1,000 to do a GWAS, how are these companies offering a product for $59?
Yeah, so this is, there's different technologies. So this is a whole genome sequencing. These, where you get actually the full genome, like every single base pair. The direct-to-consumer kits usually are just genotyping arrays, and there you only get like a handful, like 100,000 or a few 100,000 variants. So those are normally much cheaper. Like we can get them for like $50. Okay. And then some of these companies actually also provide these disease scores. And I'm not going to get into the company disease scores, but in a nutshell, what they're doing is they're providing you kind of the genome and then how many disease variants or disease risk variants an individual has compared to the rest of the population. And so how do they get these disease scores? And actually, how do we find regions in the genome that are associated with disease risk or some trait of interest? So this used to be done using linkage analyses. So we had pedigrees, and we're mapping disease risk within these families. And so the idea is, how is our disease status segregating with a genetic marker? And so this is like a cartoon example. And so you have 3 generations, and you have parents and offspring.
Pointer, can you just use the mouse? Or?
Oh, yeah,
yes.
I can use the mouse. Do you see the? Oh wait. Oh,
Lights down.
oh, okay.
So,
Should we turn the lights down?
Yeah, see if you can take the lights down a little.
I learned something new today.
High tech.
sorry, I should have done that earlier.
And so, yeah, so you have the 3 generations, and then you have like offspring, males and females, and then the filled in shapes are those that are with the disease. And so you can see across the generations, if you look at which marker is segregating with disease, we see that the B2 allele tends to be present every time an individual is affected. So these linkage analyses are targeted for these variants of high effect and that tend to segregate within families.
So if you have a gene like CF and there's 600 genetic variants, could you have like a package and you just do all 600 of those? Because most of the gene tests for CF now, they give you like 12.
Yeah, so I think it's come quite a long way where they figured out which variants are likely to be the causal ones. But when we initially do the analysis, you'll see later it's quite a large region, and then we have to do a lot of follow-up analyses to really narrow down which are the causal genes, or which one is a causal gene potentially, and which are the variants that are implicated. Yeah. So it depends on like do you want to test it in a clinical setting, or do you want to find new associated genes? Okay, and like we mentioned, one of the big wins from this was finding the CFTR gene in cystic fibrosis. This is another example. This is a linkage analysis done for the skin prick test, which the study was using it as a proxy for atopy. And so this is what a linkage analysis result would look like. So on the x-axis, you have all the different chromosomes and the different regions they're testing, so they binned it. And the y-axis, you have the minus log 10 p-value for how significant is this association or this region associated with this skin prick test in these families. And so you can see there's a hit on chromosome 17 here. But if you actually look into that region, it's quite large. It spans 28.5 megabase pairs, and that is including over 100 genes. So there's a lot of follow-up work that is involved to try to narrow down what is, like I mentioned before, the candidate gene in that region. So kind of getting into how GWAS came about and why there was a need to go from linkage to GWAS and what allowed us to get there. So linkage really is good for mapping within families and finding these variants of large impact. But as I showed you before, these regions that they're finding are really quite large. And why that's the case is if you think of within a family, if you have the genome of like the mother and the father, over time, even within a few generations, the variants that are inherited together are going to be broken up by recombination. But only after a few generations, that region isn't going to be broken up enough. So you still have these large blocks of variants that are inherited together within the family. The other idea of why we wanted to move from linkage to GWAS, and I'll show you what GWAS is in a bit, is this idea that when we're looking at families, we're looking for these variants of high impact. But if you think of common diseases and the genetics that are affecting these common diseases, you wouldn't think that their frequency is high, because if they have a high impact and a high frequency, selection would likely reduce the frequency. So the idea would be if you're a common variant, the more common your frequency is in the population, the smaller impact you have on disease risk due to selection.
Somebody, please mute your phone.
Okay. And so how GWAS work, it really banks on this idea, as I mentioned, that over time, over generations, recombination is really going to break these, what we call linkage disequilibrium blocks, or LD blocks, blocks of variants that are inherited together. And so, within a family, these blocks are going to be quite large. But over time, across generations, thousands of generations, you're going to have blocks that are tinier and tinier and tinier. And so this helps us when we look at GWAS. It's basically linkage analysis, but in unrelated individuals. Now you're looking at variants that are overrepresented or underrepresented in disease individuals. And these blocks that you're going to map them to are much smaller than what we would see in a family setting. So what we do is we normally get a variant that, so if you think about an LD block, and we have a disease risk SNP, so this is the causal variant, what we can do is we can get any of the genotypes for the SNPs in this LD block and use it as a proxy or a tag SNP for the disease risk variant. And then I'll show you a little bit of an example later on, but we look at whether the frequency is associated with being a disease risk or not. The very important note about this is that these LD blocks are going to be different across ancestries due to population history. So how this impacts when you do the GWAS is that if you have a SNP that you think is tagging the causal variant in one population, it may not tag the causal variant in another population. Another aspect of this is that, in African ancestry, these LD blocks tend to be quite smaller, but in Europeans, these blocks tend to be larger because the European populations went through a bottleneck. So this actually caused the LD blocks to be quite larger.
What does that mean?
Yeah, so this gets into the nitty-gritty of population genetics, but it means that if the out of Africa model, so when the European population moved out of Africa, there was only a smaller number of individuals, and so there was less genetic diversity, and so that's why the LD blocks are much larger than the African population, which stayed and they were able to maintain the larger number of individuals and genetic diversity.
If you go back a little bit to the slide, we had skin test results and linkage.
Yeah, this one.
So I don't quite understand how you generated that data. You got all the chromosomes this way. What's the log transform p-value mean?
Yeah, so they basically do this transmission test where they look at, in this, let's say, for example, this B2 allele, how many times is it showing up in a diseased individual? And so there's a metric there. And then you get a p-value from that statistic, and that's what they're plotting here.
So how is the skin prick data, where is it in that analysis? Positive or negative? How many or?
Yeah, so.
Just in a clinical way, where did that data get assembled into that graph?
So I think they did, so what happens is basically you have, let's say this color would represent the skin prick test. So they had a positive skin prick test. So that would be a filled in square or circle. And then they would look at the allele and how it segregates with this positive skin prick test. Okay. So getting into genome-wide association studies, so now instead of families, we're actually looking in these unrelated individuals. And so what we have are a large number or a number of cases, so people, let's say, with asthma, and then we have a large number of controls, people without asthma, and then we look at how these variants and their frequencies are correlated with that status of having asthma or not asthma. So in this toy case, we have this red variant or red allele, and we see that its frequency is much higher in cases than in controls. And so you can do a test to assess whether this is significant or not. And so that's, in a nutshell, how we carry out GWAS. Okay. So what do we see from these GWAS, and what kind of results are we getting? So this is an example. It's quite small font, but you don't just ignore the genes for now. This is actually carried out a few years ago, and it's the largest GWAS to date on allergic diseases. They had 180,000 cases and 180,000 controls. And so what you're seeing here is what we call a Manhattan plot, but it's actually flipped on its side. But if you imagine it kind of rotated, we call it a Manhattan plot because it's like the buildings are the skyline in Manhattan. And so what we have here is now on the, I guess, the y-axis, you have all the regions on the different chromosomes, and on the x-axis, you have how significant is that variant associated with case status of having allergic disease. So basically, we're going back to this test, and we're testing at each variant, what is its frequency, how is it correlated with disease status? And so there are actually really large number of regions and candidate genes that are associated. There's about, you can't see this, but it's like about 50 regions that are, that they found that were previously reported from other GWAS studies. And then there's another 50 that they actually found that were completely new that hadn't been discovered before.
Well, what's the definition of allergic disease?
Yeah, so they said history of eczema, asthma, or atopic dermatitis. Wait, let's see.
Is that from patient reported or physician reported?
It's physician reported, but they also had.
That doesn't make it right.
But I think it's different if you're doing like a survey and they say, oh yes, I have asthma.
Yeah, yeah. It
Versus.
should be better than patient reported.
Yeah, so actually, there are a fraction of the subjects that are 23andMe subjects. So those are self-reported cases. Yeah, so what's encouraging is that some of the 23andMe GWAS that they've done, when they compare it to really well-curated GWAS cohorts, they do see replication of the known hits. So yeah, they do quite a bit to make sure, at least for some of the phenotypes, it's decent. Some of them are really bad. Like if you try to look at RA or lupus in 23andMe, it's self-reported just doesn't do it.
Two quick questions. When you're talking about like these arrays, the genotypic arrays, like what is the coverage on them?
Yeah, so there's been a lot of work to try to get full coverage, at least all of the LD blocks.
Okay.
So It's about usually ranging from 500,000 to 700, 800,000 variants. But
Okay.
what they can do is they have these reference plans where they have done whole genome sequencing or on a smaller number of individuals of similar ancestry,
Right.
and you can use the pattern between variants to impute.
Please mute your microphones.
Or stop flushing the toilet.
You can impute using the patterns you see in the reference panel.
Uh-huh.
You can impute untyped SNPs.
Okay.
So as long as you
Yeah.
have good coverage across the genome, you don't have to have all the variants and you can
Right.
impute it. One problem with that is that it's only common variants. So
Right.
these are only variants that are more than about 1% in the population.
Right.
Once you want to get down
I
to
mean, it's an inherent issue with SNPs, using
Yeah.
SNPs to,
exactly,
Type, right.
yeah.
What's the
Okay.
definition of the word coverage here, and how much of the genome you actually have sequenced?
no, it's
No.
the number of replications you look at the gene with. So it's not, because it's the number of times you basically amplify it in the sequence to look at it.
Oh, sorry, I misunderstood the question.
Okay.
then. Yeah, so.
I don't even understand the answer.
So if
That's why I was wondering when you said 500,000.
Yeah.
Is that about base pairs or?
Yeah.
You were talking about base pair like numbers.
base
I'm talking
pair
about
coverage.
how deep.
Yeah,
Sorry.
so genotyping arrays, we don't typically look at them with like coverage in that
Right.
sense, because when you do whole genome sequencing, you're actually getting these reads that map to the genome.
Right.
And there you can think of coverage where, how many times are you seeing something from this region?
Right.
Yeah, and so genotyping arrays, they kind of do a different, they
It's a
look at
different technique.
intensity, so
Okay.
it's a different technology.
Yeah.
Yeah.
Sorry, just the second question really fast. Your x-axis, is that,
Yeah, so the x-axis
Is
is
that
the
just
minus
log?
10 p-value.
Okay, good.
Yeah,
All
and I actually I forgot to mention the red line that we see is actually, so we are testing a lot of variants, and so we have to test for, or correct for multiple testing, and so that red line is the, what we think is significant after we've corrected for all those multiple tests.
The red vertical line.
Yeah.
So everything to the left of that, which is like Manhattan skyscrapers,
Yeah.
is important.
Yeah, exactly. The rest could still be true, but you might also enrich for false positives.
So when you get these gene regions, how close can you focus down? Can you actually eventually go down to a single gene and say, this is what we're looking at?
Yeah, exactly. So I don't get into it in this talk because I could fill up a whole other hour talking about this. But what you do is you have these variants, and then you want to ask, are they any coding variants for genes? Because when you do GWAS, a lot of these SNPs actually fall into intergenic regions or intronic regions, so they don't quite map to a single, well, intronic, yeah, but intergenic regions, they don't quite map to a single gene. Classically, what we've done, which is not right in the field, is just tag the closest gene. But as more data has come out on how genes are regulated, we can see that SNP variants that are much further away from genes can still regulate them. And so what we do is we look for coding variants, but what we more often do is actually look if any of these variants are also associated with the expression of genes. And so then you can narrow down to, this is the likely candidate gene. And that's actually what they've done here, but yeah, I've kind of hit that data. Okay, so you have these 100 regions that are associated with disease, and a lot of times when we bring it to our discovery scientists or discovery biologists, they're like, well, what do I do with all this? Like, I don't know. So sometimes you can look through all the genes, see what are interesting, but another thing that you can take this data and do is you can ask, okay, I have 100 regions. What do these regions have in common? And so this study actually found that if you look at the genes that are near these regions associated with allergic disease, they're enriched for expression. These genes are enriched for expression in the blood and lung tissues. And if you take it one step further, you can see that these genes actually are enriched for regulating regulatory regions in immune cells. And so what often we do now in the field is we take these GWAS hits and we use them to determine what are the most relevant tissues for our diseases or traits of interest. So for here, the study suggests blood, lung, and immune cells are really important for allergic diseases. As we get more data, so now it's usually bulk tissue that we have, but once we get more into single cell data, we can really hone in on the really important cell types that are implicated in disease risk. Okay, so that was allergic diseases in general, kind of.
Does that mean that, like, if you have a certain SNP, it's not expressed in every cell in the body? It's only expressed in respiratory tissue?
So it's the SNP, that's the gene that it might be regulating. So that gene might not be expressed in all tissues, or it might be more highly expressed in one tissue over the other. Yeah, because here they were looking at genes. And then the second part is that SNP is in a, let's say, like a methylated region that is only unique to one type of immune cell versus another cell type. So this is the Manhattan plot for GWAS that was carried out on general asthma. So these are cases that I think these are actually well curated cohorts where academic institutions have gotten a number of asthma cases, and this is general asthma, so from mild to severe, and they carried out a GWAS on 24,000 almost cases and 118,000 controls. And they also found a number of different regions that were associated with asthma risk. But one question that kind of comes up in the field is there's different severity levels in asthma, and so one might ask, does genetics contribute to whether someone becomes, not only gets asthma, but has a more severe version than another individual who only ever gets mild asthma? So last year, now, I guess we're in 2020, a study was published on looking at the risk of moderate to severe asthma. And so here they had a number of different cohorts, one of which was obtained from a biobank. And so what they had was, they said these individuals must have asthma, but they also must have moderate to severe asthma based on this British Thoracic Society guidelines. So they used, basically most of the guidelines have to do with use of corticosteroids, or, and so what they have is two different cohorts. So this was their discovery cohort, and they try to replicate their what they saw in this replication cohort. And you can see a number of different metrics for these moderate to severe asthma patients. And what's kind of encouraging is that you see that the FEV1 over FVC ratio is lower in the cases than controls in both cohorts. So when they carried out this GWAS, they also found a number of different regions. Now they've labeled it with the candidate gene. But I think what's more telling is if you put this Manhattan plot against right next to the one that I showed you previously for all asthma, and you can see that most of the hits are actually shared. So this HLA region is shared, this chromosome 17 region is shared. It's different levels, but if they actually went and tested every SNP that has been associated with asthma in their moderate to severe asthma cohort, and they saw that most of them are replicated. So what this suggests is that, genetics may play a role, but it's probably the environment that plays a larger role in determining who has moderate to severe asthma versus just mild asthma.
Taking this one other place, could you take a disease that we're trying to find an antibody for, like a melanoma or a glioblastoma? Could you actually take uninvolved tissue and involved tissue in the same person and find out which genes are different? In a disease like that, where you might be able to find a monoclonal marker that you could actually use to tag those tumors?
So looking at like differential gene expression?
Yeah, like a melanoma, send it right next to it normal skin and just see which gene is changed the expression.
Yeah, definitely. So a complementary analysis to this. So here we're looking at variants, but what we are also interested in when we get to the gene level is looking at, you can do it in melanoma, like tumor, non-tumor, or affected, non-affected, but you can also do it in asthma, looking at expression in the lung or epithelial cell, lung epithelial cells, the expression of certain genes in asthma patients versus healthy controls. And you can also look at the differential expression of genes in that way too. So that can complement this analysis.
Cool.
Why does this, these plots lead you to the conclusion that it's largely environmental factors?
Yeah,
What
so
is that? I mean, you plotted genes and shown it appears that certain genes clearly have a clinical significance, whether it's mild, moderate, or severe.
yeah.
But I don't see how you draw the conclusion that it's largely environmental.
Yeah, so the idea is that if it was genetic, then we would see many more genetic variants that are only associated in moderate to severe asthma. And so it's hard to tell a little bit from these plots, but many of these hits actually that they see in moderate to severe asthma, you can find associated in mild asthma. And so the idea is if it was really genetics that was distinguishing, that was making someone become a severe asthmatic versus mild asthmatic, you would see these hits that are enriched in one type of asthma versus the other. But we don't. What we see are a lot of the hits that are just associated with general asthma risks.
Well, I would think that the question we want to ask is asthma different than non-asthma? And not the distinction you just made, that mild is different than moderate and severe. Just a yes/no answer. Do we see consistent genetic abnormalities in people with asthma versus control population?
Yeah, and I think we do, right? So these are all genetic variation that is different between an asthma patient versus a healthy control. Why we were interested in if there was any genetics associated with moderate to severe asthmatics compared to healthy controls and not seen in all asthma is that these severe asthmatics tend to be uncontrolled. And so if you want to find a target that is unique to them, you might have more power in looking at only severe asthmatics versus just looking at everybody. But from this analysis, it looks like there might not be that many unique genetics. Like they did see one hit, the MUC5AC on chromosome 8, that was only associated with moderate to severe asthma and not in all asthma. But generally, all of the lesions are shared across the two.
And have they ever lapped information from like mouse asthma models over this to see experimentally induced asthma are any particularly good predictors, for testing?
So could you
With regard to the different
so if they
genetic analyses.
if they overlap kind of experimental data from mice on these genes to see if any of them are also have a phenotype in these. Yeah, so I think there are some that have done this, but it's usually not in the GWAS papers. It's usually like independent studies that take these GWAS hits. So I think there's been some done probably for like IL-33, ST2, probably IL-5. There might be mouse studies as well, but in these studies they tend to just look at the genetics. But at Genentech, that is something that we're interested in because genetics. These GWAS studies can only get you so far, and we want to confirm that the candidate genes that we're thinking are associated with disease risk are showing a phenotype in mice, and like what are these genes doing? So the mouse models can help quite a bit with that.
If you look at the genes here that seem to be at the highest bars, so to speak, do they have, they make clinical sense? Are they genes that you would expect to have a biologic function in causing asthma?
Yeah, I think it's funny. So HLA makes a little bit of sense, right? It's associated with a lot of immunological phenotypes. The one on chromosome 17, so that one is, the candidate genes are gasdermin B and ORMDL3. And there's a lot of debate in which one is the real, causal gene, but there's a lot of hypotheses on how they influence asthma, but it's not obvious. Yeah, exactly. I think there has to be a lot more work done on those genes to figure out what their role is in asthma. A lot of times what's difficult about these GWAS hits is they bring up genes that not a lot is known about them. Like if you ever get an open reading frame gene like C or blah, 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 that's like, nobody will touch that because it's like it's a lot of work to figure out what this gene is doing and then what is this gene doing with respect to asthma or your phenotype of interest. Not to say we don't, but it just takes a lot more work to get there. Okay, so I've shown you examples of GWAS carried out in asthma and allergic disease. What can we do with this? So as I mentioned, we're really interested in using genetics to help our target discovery pipeline. So as you know, a lot of clinical trials fail due to efficacy. And so what this study showed, I think these were from another pharma, I don't quite remember which one, but they took a lot of these targets that were, and then which phase of clinical trial they were in, from preclinical studies all the way to this target was or this drug was actually approved, and they looked at how much genetic support these targets had in each category. So what these categories are, so OMIM is a database of genes that are implicated in Mendelian disorders. GWAS-DB are genes implicated by GWAS associations, and then these are both of the databases put together. And what they saw was that as you got further along in the, clinical stage of a drug, they were more enriched for having some sort of genetic association with the disease they were targeting or a similar related trait. And so this is encouraging, suggesting that, we don't only ever look at genetic evidence to support going forward with a target or not, but having genetic support seems is a good, good figure to have in addition in your portfolio of whether or not to target this this gene. And so I just brought up a couple examples. So for example, there's a I think phase 3 now, clinical trial for TSLP, tislelizumab, and there are a couple trials going on for IL-33 and its receptor ST2. Okay, another thing you can do with GWAS is you can actually look at which traits are related to each other, and so this is what we call genetic correlation. This is the fraction of genetics now, genetic risk factors that are actually shared between two traits. And so in 2017, this group looked at asthma, and actually they were doing a COPD GWAS. And they saw that actually asthma and COPD had some shared genetics. And so this is from there. So what you have is COPD and how much genetics it's sharing with these different traits. So these are lung function traits, asthma, and some other BMI related and height related traits. And so this says that about 38% of the genetics is shared between asthma and COPD. And what the color represents, or the direction, is that it's saying the genetics or variants that are increasing someone's risk of COPD are also increasing the risk of asthma. So that was really interesting. And we started thinking about it a little bit more. And we're thinking about other risk factors that were present in asthma. And so we knew quite a bit that immune cells, especially eosinophils, are really crucial to asthma. And so we were wondering, what is the relationship between all these different traits? So what we did was we took the asthma data, the moderate to severe asthma GWAS that I had showed you previously, and we asked what was the genetic correlation between asthma risk and lung function, all these different lung function traits. And you also have a GWAS on blood eosinophil levels. So these are looking at the genetics of, eosinophil blood count in the blood, eosinophil count in the blood. And what we see here is that asthma and lung function are generally negatively correlated. So that means genetics influencing or increasing your risk of asthma or reducing various lung function traits. On the other hand, genetics increasing your risk of asthma are increasing the blood eosinophil cell count. Okay, but how does eosinophil and lung function relate to each other? Is this all pointing to the same genetics, or is it also pointing to different genetics?
So does that data mean there's a strong positive correlation? If you have a high blood eosinophil count, you're likely to have asthma? And converse with the pulmonary function?
Yeah, so this is just genetics. So for example, here with COPD and lung function, you see that they're almost 90% correlated. So that does suggest there is a lot of genetics that is contributing to both factors that are shared. If it was 100%, then I think you could make that, if it was 100% genetically correlated, let's say, you can say that the genetics influencing both traits are completely shared, but I guess it depends on what fraction of the phenotypic variance is due to genetics. So you can, for example, have a situation where, let's say you have 10% of the phenotypic variance is due to genetics and 90% is due to environment. And then if that 10% is shared with another trait, you still have that 90% of environmental influence.
What are these correlations?
So these mean that of the fraction of genetics that is contributing to asthma,
Turn off the CD.
and of the fraction of genetics that is contributing to lung function, they share 30% of that. So there's still, or 36% of that. There's still about 60-something% that is not shared.
And what about the eosinophil?
So this one is about 50%. So 50% of the genetics that influence asthma also influence eosinophils.
Why are some negative and the eosinophil one not?
Yeah, so this means it's the direction of the effect. So this, for example, in the eosinophils, this is saying that if the genetic variants that increase your risk of asthma are actually also increasing the likelihood that you'll have a higher blood count. But it's not fully determined by genetics yet. Okay, and then we also have here looking at the eosinophil blood count now and its genetic correlation with lung function. And we see that it's generally fairly low, suggesting that these really represent distinct biologies. So the biologies that eosinophils, I guess the pathways that eosinophils are going through to impact asthma risk are distinct mostly from the ones that affect lung function.
Is there any correlation between a genetic correlation and a p-value? Because I think that may be where we're getting confused.
Oh,
That's not a p-value.
no,
that's
it's
a
just
separate-
like an R squared, yeah.
All right.
So I guess the question is, when they were choosing the asthmatics, were they, right, because there's different phenotypes, are they looking at all comers or are they looking at?
So this one was moderate to severe asthma in their asthma GWAS. The lung function ones were just healthy individuals.
So there wasn't, they didn't break the asthmatics down by the actual phenotype, it just went by, let's pick that, okay.
All right, now we get into the disease risk scores. And so this is somewhat what we're starting to see in the, I guess, the mainstream, these designer kits, or sorry, direct-to-consumer kits. And so what are polygenic risk scores? These are basically, let's say you have all these variants that you know are associated with asthma risk, and you have individuals, and you know what variants they have. So you can tally up these variants, and you can give each individual a score of how many disease risk variants they have. So for example, the first orange person has 3, and the last person has 2. And when you have a population of these individuals, you can actually see a distribution of these risk scores. And so what we would say is that these individuals on the far right have a very high genetic risk of getting this disease. And the individuals on the left have a very low genetic risk of getting the disease. These polygenic risk scores give you, you can use them in many different ways. For example, you can say, in these high-risk individuals, even though they have a high genetic risk of getting a disease, if they don't have the disease, what are the factors that are protecting them from getting the disease? On the converse, you can have individuals with a very low risk of getting the disease, genetically speaking, but they have the disease. So what is making them get the disease? What are extra risk factors? So how we use polygenic risk scores was we actually used them to look at what factors were influencing the age of onset in asthma. And so what we have here are for 2 different data sets. So we looked at our internal clinical trial data set across a number of different trials. these moderate to severe asthmatics. And then we also looked in a biobank data set, and I'll go into that a little bit more later on, what the UK Biobank is. And we looked at the asthmatics, and then we scored individuals on how likely they were to have allergic disease, asthma, high eosinophil blood count, and then different various lung function parameters. And we asked, given these individual scores and asthmatics and healthy individuals, what were these scores for a risk of allergic disease associated with age of onset? And so if you have an effect that was less than 1, this would mean that that phenotype was associated with younger age of onset in asthma. So here, younger tended to be less than 12 years of age. And then if you're above 1, it said that this phenotype was associated with having an older age of onset for asthma, like over 25. So what we can see is that for allergic disease, asthma, eosinophils, they tended to be associated with younger age of onset, meaning if someone had a higher genetic risk for allergic disease, they were more likely to get asthma at a younger age. And on the converse, if they had genetics that was improving their lung function, they were more likely to get asthma at a later age of onset. Okay. So in the interest of time, I'm just going to skip over this a little bit, but just to say that when we create these genetic scores, we have to be aware of genetic ancestry when we carry them out, because that can bias our results. Okay, so in the last 15 minutes or 10 minutes or so, I just want to chat about where we're going forward from here. So I've shown you a lot about disease risk genetics, but a lot of times when we're carrying out these clinical trials, we're not looking at whether or not someone gets a disease, right? That's not the clinical trial endpoint. We're looking at, for example, in asthma, how many of these people have exacerbations. And so while the GWAS field is really great in looking at disease risk genetics, these progression phenotypes, these disease subtypes or disease-related phenotypes are a lot harder to come by. And so now what we're trying to do in our department is really try to focus on these clinically relevant phenotypes. So how do we get that kind of data? We can look at our internal clinical trial data. These tend to be really well characterized samples, and they have rich phenotypes, but the sample sizes tend to be small. So it's going to be really hard to get to those like 10,000, 100,000 samples that we saw previously in those GWAS. Another way to get about it is via collaboration. So we often collaborate with academic groups. And these also tend to be well-characterized samples, but the sizes of the cohorts and the phenotypes available tend to be variable. In more recent years, a lot of these biobank cohorts have been coming out. So these are cohorts where they have linked electronic medical records, and the sample sizes of these cohorts tend to be quite large, but the problem is they're less well defined. You have to use ICD codes to define your relevant phenotypes, and I'll show you a little bit what that means later on.
Well, those of us who do EMR, we know what that means.
Oh, yeah, it's true. You know much better than I do.
Garbage out.
So this is just a quick overview of what the UK Biobank is. It's quite a large cohort, like 500,000 samples, and they really targeted individuals that were in their middle age and relatively healthy, because what they want to focus on is how these individuals progress over time and what influences their risk. So this is really like a longitudinal cohort of massive size. They have linked electronic medical records. Every time these people go to the hospital, you have all that data, all their patient records, like what procedures they went through, what ICD codes they got tagged with, how long they were in the hospital for. They also have really extensive baseline questionnaires, but they also have baseline sample measurements. So they have like lung function measurements, they have blood samples, they also have a number of different imaging metrics. So this is a pretty powerful cohort with a whole host of different phenotypes available. And they also have genetic data available, hence why I'm speaking about it. So you guys, of course, are much more familiar with this than I am in the ICD code space. So this is kind of what the EMR data in the UK Biobank looks like, or just a snapshot of it. So for example, this is how many people, the number of individuals who went into the hospital for these various Chapter 10 respiratory symptom ICD-10 codes. So you can see if I was interested in asthma, I could, for instance, look at J45. So what we wanted to do with this cohort is that we have all these individuals who are going into the hospital for asthma. We wanted to look at exacerbations. But the question is, this is an EMR cohort, and we're not sure how accurate it is looking at these kinds of phenotypes as compared to a well-curated cohort. So what we did was we wanted to just ask a simple question and see if this data could be reflected in the phenotype we're interested in could be reflected in this data. So this was published a couple of years ago, and I think we all know about the seasonality of asthma and exacerbations. So just focus on the placebo arm for now. Actually, just focus on the placebo arm. So we see that there's a peak in exacerbations in the winter, and then there's kind of another peak in the fall. So our question was, can we see the similar pattern in UK Biobank? And if we can, then we can go forward and really dig into that data and the genetics of exacerbations. So I mentioned this previously. These are all the kind of hospitalization episode records available for the patients. And then these, because I wasn't familiar with ICD codes before working with this cohort, I had to look up what codes were available for defining asthma. And please let me know if these are inaccurate or there are other codes that are missing that I should be using.
They use the same codes in the UK?
I guess so, yeah.
International.
Huh? It's international?
ICD, yeah. It's supposed to be.
Okay, so I had the codes, I had the data, so I pulled out how many hospitalizations an individual had over the time they were in the biobank, or they had records. So what I'm showing you here is the number of samples and the number of the number of exacerbations per sample and the number of samples with that exacerbation. So this big bar are all the individuals who only had one hospitalized exacerbation due to asthma. And so I looked at this, there were about 6,000 hospitalizations across 2,600 individuals. It was a bit strange that I found this person who had 108 hospitalizations. When I spoke to a clinician at Genentech, he kind of jokingly said this is insurance fraud, but it's the UK, so yeah, I don't know. But when I looked into this, the records for the sample, I realized that they had been going to the hospital, leaving, and then coming the next day and leaving again, and then going the next day and leaving again. So those technically weren't independent exacerbation events. I'm not sure why they kept going and coming back.
Insurance fraud.
Insurance fraud, yeah. But what I did was, okay, I realized I had to QC the data a bit more and require that the individual was hospitalized for at least 1 day and at least 30 days apart. And there I got much more reasonable data. There were about 31 hospitalizations across 2,900 individuals. But most individuals only had one exacerbation. Okay, so going back to our question from before, do we see a seasonal pattern to these exacerbations? So we do. What we see here is, okay, the exacerbation rate on the y-axis and then across the different months. And so what you can see is that there is a higher exacerbation rate in the winter and the fall, and it's the lowest in the summer. Okay. So another tidbit that the study found was that if you divide the patients into individuals who had a high eosinophil cell count and those who had a low, only the individuals with the high eosinophil cell count had the seasonal pattern, but those with the low ones did not. And so because UK Biobank had blood CBC data at baseline, I could actually take the eosinophil cell count of these samples and divide the patients into high and low eosinophil cell count samples as well. So this is a similar plot as I showed you before, but now I just collapsed the months into seasons. And so in red, you have the exacerbation rate for EOS high samples. In blue, you have the rate for low. And what you can see is that, well, we do see something that that eosinophil high patients are exacerbating at a higher rate, but we don't see that the EOS low samples don't have a seasonal component to their exacerbations. So one thing to note is that, the previous data I was showing you was a clinical trial cohort, and the exacerbations they were looking at were not only hospitalizations, but there were also individuals who had worsening symptoms that they needed corticosteroids for. So this isn't captured in the UK Biobank data, where I only looked at hospitalized exacerbations. So once we, if I can find pharmacy data, that might be something we can add to this as well. Okay, so that concludes it. So just to summarize, I told you a little bit about how we're using genetic associations with disease risk to guide target discovery. I told you a little bit about GWAS can help find genetic vectors and show you how there are related diseases, genetically speaking. And then finally, showing you about kind of in the future, what we're looking towards is using a lot of these electronic medical records to help us further our genetics beyond looking at disease risks. And then this was back to the word of the day, but I'll just leave it at that. Thanks.
I have two questions for you. One, a broader one. How do you use this to help you in drug development? So if you knew all this, how would you design a prototype drug, to be the cure of asthma? That's one. And the other question would be, let's say as a proof of concept, GWAS, if you took a disease like sickle cell disease, we know the answer ahead of time, you know exactly what the genetic defect was, and you did GWAS, would you have a perfect correlation? Would you find, using genetics, you would diagnose this as a 100% genetic disease? Anybody done that?
Okay. So for the first question, how we're using it to design the cure drug. So I think we're pretty far from looking. So I'll just bring up so when you see these GWAS, yeah, this is a good example. So this is just the first step, is finding the regions that are associated. And what we do is we next speak with our discovery biologist colleagues who are really taking the looking into these animal models, looking into how the genes function and how we can actually get a target from them. And so I bring this to them and I say, look, these are the regions. Maybe one of these genes are really interesting. Can you go forward and do some experiments to figure out what cell type of interest, what tissue it's working in? Is it relevant to phenotypes that you're interested in that you think are important for asthma? And then it kind of goes from there. So I think genetics is, it's kind of shining a flashlight on the genome and saying, oh, hey, pay attention to this region. And then there's a lot more work that goes on after that to really determine whether or not we should go forward with that.
Has this led to any drug study yet in humans?
Yeah, so I think the classic example is PCSK9 in the cardiovascular space. The asthma examples I showed you, so that I won't, the ST2 and TSLP, I mean, I'm not sure if the genetics actually led to those being put forward, or genetics was the reason that they were carried forward. But I think in the future, more of these genetic hits are going to be used, and more of this genetic data will be used to feed into our drug discovery pipeline. And then the second question was...
Second question is, if you took a purely genetic disease like sickle cell disease, and did a GWAS study, you should get a probability of 1.
Yeah. So
does that work?
Yeah. So what, and actually what I don't speak about here are the clinical panels that people can use to determine risk of these variants that we know are medically relevant. So for example, there's the BRCA1 gene for breast cancer. There's ApoE4 for Alzheimer's disease. For asthma and allergies, that isn't that much. But if you were to carry out 100% all the individuals with this variant have the disease and all the controls don't, if you were to do the association, your p-value would be through the roof probably, because you just have 100% distinction between the two.
Well, it should be. I mean, it'd be a proof of concept that it's worth doing this work.
Yeah.
That it gets you an answer.
the association of the asthma and eosinophils, it didn't appear that the eosinophils related to lung function. Is that correct?
Yeah.
Have you been able to track it to other aspects that where the eosinophils are implicated?
Yeah.
I mean, did you look at methylcholine reactivity or airway hyperresponsiveness or
Yeah, so those are
cell
phenotypes
trafficking?
we're really interested in, but unfortunately, a lot of the GWAS are fairly small. So to do those genetic correlation analyses, you need sample sizes that are at least 5,000 samples. So the methylcholine challenge, GWAS tend to be more on the order of hundreds of samples. So once we have more sample sizes, we'd love to dig into that phenotype more and look at where the eosinophil and also the lung function, where that's really mapping to.
And have you looked at that or others in other biomarkers, the asthma with like exhaled nitric oxide or periostin levels?
Yeah, so I think we have in our internal clinical trial data, so we should probably take a look at that, but I haven't taken a look at it yet.
Well, the data you showed was in the United Kingdom.
Mhm.
Are you doing something in the United States with any one of the big healthcare companies, or how does it work here?
Yeah, so there's no slide here, but there are a lot of big biobank cohorts happening in the US as well. So there's Geisinger, and then there's one from the VA. And so working with the VA has been a little bit harder. We've been trying to collaborate with them, but I think now we're going through other routes to get access to their data. But also it's not as standardized in the US, so it's a little bit harder. Yeah.
I remember at a national meeting, I don't know, maybe 5-7 years ago, somebody came out with, I think, the ADAM33 gene as being supposedly the prototype gene for asthma.
Mm.
You know
That was
that
40 years ago.
Am I right about that?
That was
Remember
40
that?
years.
Wasn't that a beta receptor?
When I was in training, is they came out with ADAM33, that was
Yeah.
the asthma gene.
Did that hold up?
No, it's not on any of the target. Just to bring up the dark side, how long do you think it's going to take the insurance companies to start using this to screen patients for insurance
Risks.
policies,
Yeah.
for risks and medical conditions? And what do you think of the ethics of this as it moves forward?
Exactly. So, I mean, this is just personal, this is like nothing to do with like genetics views, I guess. But yeah, I mean, I don't know, but that's something, in the field that we chat about a lot, because it is, I don't know, I feel like insurance companies are just going to want to make as much bank as possible, and there's like, pre-existing conditions, and then there's like, oh, you have like APOE4 variant, you're very likely to get Alzheimer's disease, so maybe we don't want to, or we want to increase our premium. I don't know how long before they're going to take it and use it in that setting, but I do think it would be great if there was some regulation on it, I guess. But yeah, I don't really speak with insurance companies, so I don't know what they're thinking in the back of my mind. I guess we can know by how many people, geneticists, they're trying to hire.
Well, thank you very much.
Yeah, that was awesome. Pretty soon, you're just going to be able to do it. I'm going to tell you everything is wrong.
So you were saying people who go like the 23andMe, some of them they find criminal history?
Oh,
yeah.
About this?
It's a genetic
It's how they
forensics.
found the guy in Tacoma who
Yeah.
killed that
Yeah.
girl.
Not
for
criminal,
several
how
years.
would you be so stupid as
No,
to
they're-
send in your gene?
They're not. What they're doing is they're
It
getting
was
the
the relatives.
relatives.
Yeah.
And they find out it has to be a sibling of these people.
So if I
and then
did
they
it,
have to
if I
accumulate
did it and you had done something bad, they'd find you.
Right.